It is my pleasure today to introduce to you Dr. Kelsey Canada. Kelsey is a developmental cognitive neuroscientist focused on studying individual differences in children's development at multiple levels, neurobiology to cognition, with keen interest in environmental factors that might explain differences in brain and cognitive development. The goal of her research is to gain overall insights into mechanisms of memory and brain development in order to identify sources of modifiable risk and protective factors. Got her BA from Grand Valley State University in Michigan, and got her PhD at University of Maryland, College Park. From that, she moved to do a postdoc at Wayne State University with Dr. Anna Doherty and myself. So you're going to hear a lot of really exciting projects that she's been recently working on, and it's really a pleasure to have her.
Yeah. Thanks for letting me hang out with you guys after a great conference at DACC. So, like Noah said, I work at the earlier part of the lifespan predominantly. So, out with kids who will tell it like it is. So with that in mind, if there's anything unclear during my presentation, feel free to tell me like it is and let me know if you have a question. But today I really want to talk about how I've been thinking about the ways that we can integrate the data that is both behavioral or cognitive and neuroimaging data to answer questions about individual differences, both within our own samples and thinking about going across samples. The grounding factor being, I'm really curious knowing why across cognitive development people differ in their developmental. Because if you take, for example, just the brain, you know that the brain changes across time within individuals, and that it's systematic in some ways, but it varies in others, and that ways that it varies might impact, for example, the fact that at age 4, when you do a task, kids might remember the outline shapes that you showed them. Maybe a little bit later, say around 7 or 8, they can remember more details, like the color of those shapes that you showed them. And then beyond that, they have both the outlines and the colors, in adolescence, and then that becomes stable into adult. This is very much a cartoonized example, but here you're gaining the amount of details you can recall in tandem with other underlying neurobiological changes in the brain. And specifically for me, I'm really interested about thinking how the hippocampus, which is near and dear to many people's hearts, plays a role in this because of its different functions. But critically, the hippocampus is an area that we know is extremely susceptible to environmental factors, especially stress. But in typical populations, we often don't have these extreme examples of stressful events. So it's interesting to think about resources in this household. And then I don't have a lot of data, but I'll talk to you about the way I'm thinking about it in neighborhoods as well. So just to give you a heads up, because I like stats, this is going to be the tool belt, methodological tool belt that I bring with me today. So you're going to see a lot of structural equation modeling, specifically multi-group, so using cohort sequential designs, second order relating growth models, and integrated data analysis. And all of the imaging that I'm going to be showing you today and the derivatives are coming from T2-weighted images, which is the standard for having valid and reliable estimates of the hippocampal volume in individuals. And for these hippocampal subfield segmentations, I'm very much cognizant of the validity. So are we actually getting the best possible proxy of the underlying structure that we're interested in? So I employ the best practices in, again, image collection and quality control. So making sure where we're putting those labels corresponds to what we think has functional. And if you're interested in other methods, I talk too much about them, but I'm happy to talk about them later. And so I'm talking to you about brains and methods, but really at the end of the day, why do I care about that? It's because we think about this ability that all of us have in this room, I assume, not everyone in the world, of episodic memory. And it's this ability that helps us create who we are, whether we are the glue or ourself is the glue or the memories are the glue. I think we had a conversation the other day. But regardless, the ability to form these memories that become ourselves increases rapidly in early childhood. And we see this in lab-based tasks when we ask children and individuals about the source of information. So where did you learn that cheetahs are the only big cat that can't roar? The fine grain details. So did you see something that was pink or blue? The temporal order of events. So did you first throw a ball or did you first have a hot dog at the fair? And just the overall number of details were called. So this increased fidelity of memory representations over time. And just to give you some context, because research can be personal in some ways, this is me at age 1. I have no adult recollections from this, but it's not that at age 1 I wasn't able to learn associations or do things. They just weren't the same as what I would have now. And the same would hold true when I was 3.5. It's not that if my mom asked me, what did you do today? It's this black box in which I never formed memories. It's just that something in the underlying neural circuitry wasn't at a level of maturity, for example, until about 5 to 6 years, where I can actually recall events today that have stood the test of time. So research yourself at those childhood. And beyond being interested in something that helps form our sense of self, there's a lot of reasons why early memory development matters. One is that it's a time where we see rapid rates of change. And when you have rapid rates of change, you often have increased flexibility in that underlying process. But it also becomes increasingly important in being measured externally, beyond just your mom and dad caring about what you did that day. This is when children enter formal schooling. And it coincides when there is a diagnosis in learning disabilities. And we also know, as I alluded to in terms of environmental factors, those who have different resources in their household or neighborhood tend to have different cognitive abilities. So those who have more socioeconomic resources tend to have these better performances on the test of cognition. And if we think that there's coinciding differences in the brain and cognitive outcomes, we want to think about how interventions are also going to have the greatest impact during this time. So just like in later life, memory change matters. It also is really important earlier lifespan. So this is the top half of a slide that I've already showed you about all these different tasks that we can use in the lab that might not be the most, ecologically valid, as Louis said. And we do all these things and we do these tasks, but we really don't have agreed upon gold standard task in the laboratory of what actually captures episodic memory as we live and breathe it. And this isn't surprising. You look back at work from Emil Tolving, and it said right off the bat, there's not likely a specific relation between any given task and memory process. Because tasks aren't process pure, right? They're going to require other cognitive processes. They're going to be impacted by the measurements of those tasks, whether by who is administering them or just how a person is feeling that day. If someone is more distractible, they didn't sleep, mom said no to something. And so knowing that no single task is going to give us a perfect ecologically valid measure, it becomes really enticing to think about how can we try to take away some of that variability that's not due to cognitive development and actually look at the construct of interest, which for me is episodic memory. And so one way we can do this is to go beyond the use of single tasks and use multiple tasks of memory, which I'm going to refer to as indicators. And I'll show you a structural equation model, but before I do that, I just want to introduce you to the study design that I'll be talking about the first half. So the way I'm going to be talking to you about memory development using multiple indicators comes from a sample of 248-year-old children, 100 of which are longitudinal participants. So this is cohort sequential. 4-year-olds and 6-year-olds come back when they're 5 and 6, or 7 and 8, respectively. Then we also had cross-sectional samples of 5, 7, and 8-year-olds to account for those test-retest effects to make sure that it's not just this repeated exposure to memory tests over time that's leading to improvements by comparing performance at those subsequent endpoints to the cross-sectional individuals. So in these 200 kiddos, we had different aspects, if you can think about what makes episodic memory measured, so that we could try to see what is shared amongst them that relies on that same cognitive ability. So one of these is the recall of source details. We call this source memory. So for example, kids come into the lab, they watch a video of a puppet and a person, they learn a bunch of facts, and then a week later they come in and they're tested both for the memory of that fact and also the source. So for example, I told you that cheetahs are the only big cat that can't roar. That's one of the facts that they might learn. So they would be asked, what is the only big cat that can't roar? If they are able to recall that, they would say cheetah. If they aren't not able to, they can do a recognition task. And then we say, and who did you learn that? And they can tell us either the puppet or the person, but they could also make an error in terms of saying, my mom, my dad. I think the worst I had was the wall for someone who was completely over it. But you can really get at both the fact that they're learning that fact and the source of that information. So they have to have that contextual information, very similar to episodic memory day-to-day life. We also have a measure of recognition of temporal order. So this is which came first. We call this primacy. So did you learn this first or did you see that first? They have a more fine grained measure that they perform. So temporal order. So if you show them 7 different events, for example, going to the fair or adopting a pet, can they put those events in order and what is the number of pairs that are correct? And then another binding, which is both learning where they saw something on the screen, if they saw that item, and if that's the correct location that they learned that item in. So they call this item location binding. Both remember that they saw an item and that they saw it in that location. So these are different modalities, different stimuli, different scales of time. So there's a lot of variability in the lab-based task, but really what I'm interested in is there something shared amongst them that actually gets at this underlying cognitive construct of episodic? And so that's the first question that I'm going to just talk to you guys about today. And so we can do this by asking, using structural equation modeling, if we ask, do they play together, what happens? And so what you're going to see is 4 variables on the bottom. So those ones that I just showed you, the order here is recall, recognition, feature binding, source memory. And you're going to see a latent measure. So anything that's squared is actually measured in the lab. Anything circled is not measured or latent. So it's unobserved, but we're trying to estimate that based on what we can see. And we ask if that same latent measure at 4, 5, and 6, for example, in that first set of the longitudinal design, is equivalent across time. Because you can think about when you administer tasks to different participants, you could have performance from every single person, but it might not be reflecting the same underlying cognitive processes based on how that person is doing the task, right? They could be using more working memory. They could have different attention. So in order to actually ask about longitudinal change, you have to confirm that at the latent level, you have consistency over time. And so you see if there's correlations. And so the answer here is yes. And then you have to ask, is it invariant over the entire period of time, which would be that 6, 7, and 8-year-old, because this is looking at change over the full span. And in this sample, just because of missing data, the way I did this is looking at whether the intercept of that 6-year-old group at time for their time 1 was equivalent to the late estimation of that 4-year-old time 2. So do they meet at the same point? Can you stitch those trajectories together? And the answer there is also yes. And so this is all of this background work to say you can use multiple lab-based tasks that vary in their administration, their modality, but that do share something at a late and unobserved level, which we're referring to be episodic memory ability based on the construction of tasks. But then what care about is, does that actually change over time now that we can say that these tasks actually reflect episodic memory? And so to do this, we go from level 1, so our first set of circles, up to level 2. So this is that second order latent growth model I alluded to at the first. And the way this model is estimated is piecewise, because I'm interested, if you remember I said previous work has showed acceleration from 5 to 7. So what is the amount of change between each of these time points? And is that more or less accelerated or not over time? And so to do that, you have an intercept for each of these groups. So the time 4 is the first time point for everyone here. And then you have 2 different slopes. And so you have 2 different estimates of change within the sample. So this slope between 4 and 5 is going to tell you the amount of change in the performance between 4 and 5 for this group. And that second slope is going to tell you the amount of change between 5 and 6. And then because we have that equivalency across time, we can do something very similar, but with the older kids to look at change between 6 to 7 and 7 to 8. Is everyone still with me? Okay. And so taking all of this, what it allows you to do is say at the latent level, what does episodic memory development look like from 4 to 8 years? And so what I'm going to show you here, the x-axis, you have age in years, again, from 4 to 8. And on the y-axis, you have episodic memory at the latent level. So when you use structural equation models and latent measures, it's not necessarily a reflective measure in the wild, but it's scaled to something. So here I've just scaled it to a measure that goes from 0 to 1. All right. So what we see is significant change in overall episodic memory ability between 4 to 5, between 5 to 6, between 6 to 7, and 7 to 8. So across the board, episodic memory ability, unsurprisingly, but at least consistent with all of these different tasks, increases across early to mid-childhood. But what's interesting, and what I don't have as a bullet point here, is that when you test the difference between each of these slopes, you don't actually see any difference in the rate of change. It's just a consistent increase over time, which is different from some of those other studies using singular tasks. And here I'm showing you a very clean, I have some error bars graph. But it's important to think that under the hood of all of this, that episodic memory is also marked by substantial variability. Individual differences is not the group level, it's what's different between everyone. And so if I were to pull up a figure from a different study, looking at item location binding on itself. So here on the bottom, you have age again, on each of these panels from 4 to 8. Performance here from different scales, so -1 to 3 on the first for D prime, so that's corrected recognition, and then B just have hits, and then C you have false alarms. So the amount of times kids said that they saw something when they actually didn't. And you see developmentally consistent results here, where you have increases in D prime over time, increases in hits over time, decreases in false alarms. But what I really want you to take away is how different individuals across this time period can look from each other, and just how much variability within individuals there is as between. And this is also true, it's going to be a little hard to see for sex. So here you see overlap between, people who identify as male or female, but behind the hood in those sort of opaque colors, the pink and the blue are all over the place. So some people who identify as female might have higher performance, some might have lower. It really is individual specific. And so while it's great to say episodic memory changes over time, and we know it changes with age isn't everything. And so what are additional factors that might contribute to individual differences in memory development? And an easy starting place for me, at least in this context, is thinking about the hippocampus. We know that the hippocampus is critical to memory, and that it has consistent work over decades showing that there are relations between at least total hippocampal volume and different measures of episodic memory. So what I'm showing here is a meta-analysis that I did with a colleague of mine across almost 1,400 individuals from 2.5 to 16.5. And you see that using total hippocampal volume, there's a small but positive, it pulls up there, just over from 0 here on the bottom. I have that now. Across tasks and measures. And so this isn't the same as what I just showed you, but it gives this idea that even with all this different variability in the ways that we measure memory, there's some sort of underlying correlation with the hippocampus. And this is true for tasks that are immediate, delayed, relational, non-relational. And if you had a chance to stop by my poster at DACC, you can see that with NOAA, we've shown that this is true within a sample as well, that even across these different types of measures, you can see differences in change over time. Right. But the hippocampus is not a unitary structure. Many of you were at the workshop, so you know that. It's quite heterogeneous. For those who aren't as familiar, hippocampus comes from the Latin for sea monster, which is often referred to as a seahorse in neuroanatomy courses, that has a head, a body, and a tail. And so if I were to overlay this on a structure that I'll show you in a second, so here's a 3D rendering of a hippocampus, and it has a seahorse shape, head, body, and tail, and it's comprised of different sub-functional and cytoarchitecturally distinct subfields. So you have the dentate gyrus here, which is shown in blue, the cornumonous, or the CA regions, so 1 through 3, CA4 if you want to get into a debate with some people, and then the subiculum. And I'm going to show a picture of a resected hippocampus. It's not bloody, but if you don't like that, just heads up. It's a small structure, right? So it's only, this is my hand in the picture, so it's coming across my fingers, and it's only about as big as your thumb, and you have 2 of them. So when you're looking at volumetric differences or changes in the structure, it's unrealistic to think you're going to see huge amounts of change in a structure that is already small to begin with. But nonetheless, we think they're meaningful. But this is me tempering expectations given how small it actually is. And even though the volumetric variations might not be large, they are implicated in neurodevelopmental disorders, so such as schizophrenia or even ADHD. So we think that there is some meaningful impact on structural differences of the hippocampus. And again, it's critical to episodic memory. And so before I tie this back to cognition, I just want to talk about the trajectory of the hippocampus in childhood. So there's a lot of inconsistencies in the cross-sectional research, although it does suggest overall developmental differences. And these aren't necessarily consistent because there's differences in how they're defined, which I'll come back to later. But if we sort of squint and look across the board, we do see that better memory is related to developmental differences in the hippocampus during childhood. So it serves as a potential source of those individual differences beyond age alone, and how and why individuals differ in their memory ability across this period. And so if we were to think about what the expected trajectory would be in childhood, we can look to work in non-human primates and post-mortem human studies that can look at the number of cells. And what those show is that you see the earliest maturation in terms of cellular growth, so neurogenesis of subiculum, followed by CA1, 2, and then the dentate gyrus. And then because of those recurrent collaterals between dentate gyrus, we group CA3 as not reaching maturity until about the level in terms of function. And so sort of this subiculum, CA1, dentate CA3, is what we would expect in terms of developmental change being most protracted in this bottom group, right, the dentate gyrus. So to test this, looked in, again, 4 to 8-year-olds for this part of the talk, and 167 of those 200 kids provided usable MR data at least one time point. And for kids, similar to any patient population, you have to take special consideration to get good data. So there was a lot of effort that went into this. They had a mock scanner that looked like a spaceship with a lot of feedback, and we're not above bribery. So if you're telling kids that you're taking a picture of their brain, they typically want it to look good. You know, there's a little bit of vanity there. They don't want their friend to have a better brain picture than them. So we did T-shirts, tattoos, and at time 3 for the kids who stayed in the whole study, we 3D printed little versions of their brain for them to take home. Anyways, it's effective to bribe children with their own brain. So what I'm going to talk to you now in terms of development is looking at those same subfields I pointed out before. So combining the dentate gyrus here with CA2 and 3, those are smaller subfields. On T2-weighted MRI, it's hard to get them reliably separate. CA1 and the subiculum. And for the purposes, again, of this talk, I'm focused on the body because it has the most consistency and the most agreement in terms of being able to make inferences about developmental change. I'm going to go through this part quicker than the other, but it should look similar because we're going to have indicators now instead of being different cognitive measures. It's right and left hemisphere. Don't have any hemispheric predictions. So instead, we can think about there's noise in our MR acquisitions, might depend on which hemisphere you localize to, movement, etc. And so we can say instead of averaging the left and right hemisphere, which can also average in noise, we can try to see what variance is shared amongst them and partial out noise unrelated to the actual structural estimate itself, using, again, those circles. So those unobserved constructs to say what is shared amongst right and left hemispheres gives a volumetric estimate for each individual. So we have our first order again, and then we go up and we have the same thing that we had before. This piecewise model of looking at what's the amount of change in these subfields across 4 to 5, 5 to 6, 6 to 7, 7 to 8. And that happens by, again, having an intercept. And then this slope, 4 to 5, a second slope in this group, making sure that they match in the middle, and then having that 6 to 7, 7 to 8, and again, an intercept at 6. Is everyone still with me before? And so what that lets me do then is ask at a level of using both valid and reliable measures of the subfields and structural equation modeling to partition out additional sources of variance. What does developmental change of these subfields look like in the body across this time? So again, we have h on the x, and this is going to be latent, but millimeters cubed is what it was constrained to. So that's a latent estimate here. And so what we see is that between 4 to 5, don't see significant amounts of change overall in this group in subfield volume. We see overall group increases in the subiculum and this combined dentate gyrus, CA2-3, again, 4 if you want to argue it, I have it on the slide. And then we don't see significant changes between 6 to 7 and 7 to 8. So just to recap, that dentate combination region, we see increases across this period, specifically between 5 to 6. Don't see significant overall group level change in CA1, and we see change in subiculum. And so you notice that I'm saying group change because this is similar to those plots I showed you before, where it's nice to be able to estimate at the latent level, but we also know that there's variability in how individuals actually change, which is going to become important when we talk about testing across domain relations. So if people differ in how they change over time, it doesn't necessarily matter if they follow a trajectory of the group or not. And so it's still important to ask, how does change in campus relate to changes in episodic memory with an interest in the entire time span? And so here, it's an identified model, so this isn't quite as latent because of constraints. Talk about that later if you want. Here, but you have memory on one side, the brain on the other, so this is each of those ROIs. And then for the interest of answering my questions, I want to look at, is there a relation between initial volume, so how large or small someone's initial volumetric measurement is, and the change in their memory ability. Conversely, because it's good to think chicken and egg, is there a difference in memory ability that predicts change in volume? Do you have initial volume and initial memory correlations? And then critically, for my interest and many interests, this change relation. So just how someone's brain change relate to how their cognitive ability changes over time. And so what I will just sort of bury the lead and show that although, it has this most protracted development, for in this study, we actually didn't see this correlation between changes in dentate gyrus and CA2-3 across the age examined. But there were correlated changes in CA1 volume between 4 to 5 related to changes in, this is source memory, between 4 to 5. We also saw changes in subiculum during that same period of time related to changes in source memory. And we saw that subiculum volume at age 6, negative related to change in source memory between 6 to 7. So I'll unpack this a little bit, but the first thing I want to say here is that you're probably saying there was no level change at any of these periods of time in the hippocampus, so why would you look? But the point here is, even beyond individual level, what matters is that individual variability. So the rate at which someone changed is what predicts change in memory, not necessarily that group level overall. And that gives the ability to look here. So this is variability related with variability. And so again, individual differences, we can look beyond those overall group level estimates. So thus far, on time, what I've shown you is that episodic memory can be measured as a construct using different lab-based tasks. This improves across early to mid-childhood, and that there's somewhat distinct developmental trajectories that we see at the group level in the campus, and that changes in subfield volume relate to changes in episodic memory. And although I didn't show you here, when you use a control region like amygdala, these change relations, intercept relations are specific to the subfield. But I'm still talking to you about things that are related to age. And so it's also important to think about variability beyond age in campus and age in memory. And so where I spent the last couple of years thinking is about how there are different environmental influences on hippocampal development. As many of you know, a common proxy used to think about resources in one's home is socioeconomic status. And this is correlated with both brain and cognitive development across a broad range of studies, showing that it's predictive of lifelong outcomes such as lower overall cognitive abilities. You have lesser resources, and those with lesser resources also having a greater incidence of neurodevelopmental disorder, which is harkening back to what I had mentioned to you early when we talk about Blood Hippocampus. So this isn't one of those factors that plays a role. And we know, looking at work focused on total hippocampus volume, that this link between cognition and neurodevelopmental disorders is partially explained by variability in the hippocampus correlated to these resources. But what we know is somewhat limited because most of the work is looking at variations in total volume. And so variations in both household and neighborhood socioeconomic status have been linked. Typically that lower socioeconomic status is correlated with smaller volumes, but little is known of specific hippocampal subfields. And because there are different distribution of receptors and different vulnerabilities and functions of these subfields, knowing the specific impact is very important, thinking about that connection to cognitive ability, neurodevelopmental disorders, et cetera, especially given that when you look in adults, there are specific findings correlating vulnerabilities to neural and psychological pathologies specific to hippocampal subfield that is not necessarily observed as robustly when you, for lack of a better word, mash everything together. And so working with a sample of individuals recruited from the Metro Detroit area, I looked at if there are specific relations between household socioeconomic status and subfield structure in development. So this is 167 5 to 25-year-olds, 78 of them are female, and they come from a wide range of socioeconomic backgrounds, and which provides a nice opportunity, because when I was doing my PhD in Maryland, we didn't have a lot of variability in the individuals who came into the lab. So it's hard to ask these questions if you don't have representation, which I'll talk about. So the subfields that I'm going to show you here are manually demarcated, which differs from what I showed you before, but they're still combining different subfields together. So you're going to have a combination of corneomonas 1 and 2 here, the dentate gyrus with CAB, CA3, and subiculum. And again, this is the body, because you have the most validity and reliable estimates of structure in that subregion. And similar to all of my work, I should have mentioned this on the previous slide, they're adjusted for intracranial volume. So you're not just measuring if someone's head getting bigger, is predicting these changes over time. And for socioeconomic status for these individuals, it was a composite factor looking at incomes to needs ratio, in addition to maternal and paternal education, just to not have to rely on one single measure of socioeconomic resources. And so this is a cross-sectional sample. Perhaps unsurprisingly to you, there's no robust age-related differences in subfield volume when you have it cross-sectionally for any of these. So not for CA3 dentate, CA1, nor subiculum. But what I'll point your eye to is there is a substantial amount of variability. So even though that variability is not correlated with age in this sample, there is a lot of variability nonetheless. And that variability is linked to differences in socioeconomic resources. So specifically here, we see that on the bottom, we have this composite socioeconomic socioeconomic status score. So to the left is lower socioeconomic resources. The right is higher socioeconomic resources. The y-axis, lower volume as you go down, greater volumes when you go up. And somewhat surprisingly, you'll see that those who had lower socioeconomic resources actually had larger volumes of this combination region, dentate gyrus and CA3, and that this pattern was also significant in CA1 and 2, but not in SABIC. And this is all accounting for age. So this is accounting for variations in age, even if not significant. There is a unique impact of socioeconomic resources on these subfields across this period. And I'm going to grade this out because I'll say that in addition to having statistical significance, when you actually look at the robustness of these findings, the amount of the relation doesn't differ between CA1 and the dentate, but that significance does deviate from the, so these are robust. And so this supports the idea that in a sample that is more developmental in nature, even though we're not looking at age-related change, subfields are sensitive to socioeconomic resources in the household across development. And there's a unique sensitivity in subfields that we might expect. So you have a greater impact of stress, for example, on the dentate gyrus, CA1 compared to subiculum animal studies. And although I don't have work to speak to it right now, this is likely impacting some of that cognitive function that we see differing across people with variable amounts of hippocampal change. And that highlights the importance of considering the role of environmental context, right? Individuals can be similar or different in chronological age. And so what's accounting for a lot of this variability is what's outside that chronological estimate. And when we think about where people are likely to be receptive to keeping things that are working or changing their lifestyle factors and knowing when, socioeconomic status is a place that would be right for this. It's such a broad proxy, actually being able to nail down into what specific factors would be great for future work for targeting intervention. I'm not an interventionist, but it provides this opportunity. And the last thing that I want to talk to you about is thinking about all of these things are important, I think, and they matter, but in terms of having impact on everyone, it's important to think about having these in representative samples. And because we tend to collect data siloed within our own labs, unless it's something like a PCD, it can be hard to do that. And so I started my postdoc during COVID, and they wouldn't let you near anyone with a 10-foot pole. So I had to kind of be creative about how can I ask new questions using existing data. And so what I've spent time doing is applying something called integrative data analysis to using neuroimaging. So what I equate this to is not throwing the baby out with the bathwater. So it's thinking about the fact that each study might measure something differently for an important reason. And while harmonization is a great method that we can use, it'd be awesome if we can also leverage things that already exist and are measured differently and still have meaning together. And so I'm going to walk you through kind of a dense table because I want you to think about doing this yourselves. It doesn't have to be with me. Is if you have a theoretical question of interest, you can think about if there's a team of people, colleagues, collaborators who investigate similar things to you and might have different data. And you can see if there's overlap in the representation of different constructs. So this can be cognitive, so it can be measures of memory. People do this in studies of depression and anxiety. And for here, we're trying to show neuroimaging data. And you can also think about using open data sets for something like this versus shared data. And this is the most boring part. You have to actually organize the data and make sure it makes sense. So you have to be Sherlock Holmes a little bit to make sure that you understand the data format and are able to figure out what derivative measures you would be using from each data set. And then you do the fun part of IDA, figure out what face validity is shared amongst different studies. So you don't have to have identical items, but you can look and see if there's multiple indicators, so those little squares on the bottom of each construct of interest, and if you can have them pass the sniff test. So do these two different surveys try to measure the same underlying behavior? And if so, are there questions or different parts of that that can be equated so that they can be combined to answer questions in a larger data set? And then this might be iterative conversations with your colleagues. Then you have to do visual screening to make sure that there's no crazy outliers and that there's sufficient variance. And then the critical part here is that you actually can test whether you can use items from different studies together. So you establish factor invariance, which is what I showed you slide back when I showed you all the 4 different rectangles on the bottom and asking if episodic memory is the same at 4, 5, and 6, and 6, 7, and 8. But it doesn't have to be just in age groups, right? This can be across subgroups, across sites, and like I showed you before, across time. When you test from different levels of invariance from weak to strict, and at a minimum, you have to have weak invariance, right? You have to at least have items hanging together. And if that's not met, then you can still consider alternative approaches. Still analyze data in different ways. You just can't make the assumption that at a latent level, the constructs that each study is measuring are the same. Once you establish invariance, then you're able to specify the latent factors. So this is thinking about the covariates that might be important to account for. You determine your IDA approach. I won't go too much into all of this. And then you're able to do hypothesis testing by extracting factor scores that are now, instead of being individual level estimates from each individual site, it's going to be those estimates accounting for the differences within that site. They're now comparable to test together. And this is often a continuous variable, and it's going to be specific to that integrated sample. I'll just say, if you do one integrated data analysis, it doesn't necessarily mean that you can use those factor estimates for every other sample based on who you're bringing in or leaving out of that estimation. And so just to give you a concrete example of how one might stitch data together, I'm going to show you the data that I'm using. And so far, I have the data from California, Maryland, and Detroit. You'll see there's an asterisk here next to Texas. So Texas won't be included, but just for visualization's sake of the potential full data set, we can think about representation of individuals across the United States and how there are different resources in each of these areas, but there's also different racial groups, different political leanings, different identities. And while those aren't all my questions, that provides the opportunity to ask questions across a more nationally represented sample versus just Detroit. And so you'll see here that you have this overlap that really is provided by the Detroit sample. So from 5 to 25 on the bottom. So this is that overlap I talked about. And then you can stitch in each of these other study sites because you have representation that is not solely in one location. And I'm happy to talk through all of these, but the summary is that they're approximately 50-50 on the identified sex, but there's differences in the racial breakdown in each of these groups in addition to socioeconomic. And so a starting point for me, and just to show you that it works, is thinking about using this in neuroimaging data, which is an application that had not been done before. And this is especially relevant in hippocampal subfields because there are probably at this point hundreds of different ways people have tried to measure hippocampal subfields. And so across each of these sites, there was a different protocol used to define which regions were included in a different label. So here's from College Park, this is from Detroit, and this is from Davis, California. And you can see that there's some similarity here. So there's some conceptual and larger overlap, but there are differences. So the goal of this analysis is to say what is shared amongst each of these sites, and can we estimate change in that amount of variance. So you can see here, this is a density plot of the volumes that's completely unadjusted across each of the sites. And this is for left and right intake gyres, CA3. This is for CA1, and this is for subiculum. You can see sometimes there's better or worse overlap due to the different protocols and procedures that are being used. And the idea here, similar to the work I showed you before, is that you can take these actual measured volumes from right and left hemisphere from each of them and look at what's shared amongst them at this latent level to test questions, not just within studies, but across this larger representative group. So again, just comparing speed, I was able to do this successfully. So you allow them to vary over time, and then you can add your predictors of interest. So here we're thinking about, there might be age or sex differences, there could be other covariates, and then what you get is an extracted factor score. And what you'll notice here is that the distributions aren't perfectly overlapped, but they are a lot more normalized. And the reason that there's still differences here is that there are still differences between these sites, right? You still have age differences, you still have socioeconomic and racial identity differences. And those are things that we still want to be represented in data, because if we take all of that out, we're throwing the baby out with the bathwater. So we want to keep the baby and throw out the bathwater, which is those differences in the measurement we don't care about. And so using this combined sample, what we can see even in this cross-sectional data is that a pattern emerges where we see consistent volumetric variation in dentate gyrus and CA3 across this more representative sample. And so here, hopefully you can see now that there's nice overlap in the estimation of these volumes. And here we have age and volume once again, and we don't see this in CA1 or subiculum. And there's still a lot of variability. Again, I don't have every single predictor in here. This is just a concept to say you can actually integrate these different measures of brain to ask questions and get answers like dentate gyrus does vary with age, but CA1 don't. And these were robust effects. So I'll say that subiculum and dentate didn't reach that robust significance, but you do have the effects staining somewhat. And so just what I'm doing next, I don't have data to show you, but to continue, like, why should you think about doing this? You can think about developmental change over time. So if you have different time periods, you can do this at a secondary or a tertiary time point and look at those changes now at the latent level across a more representative sample. And you can also look at predictors that modify this development, such as household socioeconomic status or neighborhood socioeconomic status, and the importance of timing. So if you think that household resources are more important early in development, which we have some preliminary data that suggests this, you can look at that actual timing and change versus if you think as dependence becomes a greater part of someone's life in adolescence, you might have a larger effect of those neighborhood resources. And just to give you an idea how someone might measure socioeconomic status, I'm just going to show you Texas. And so this is using the Distressed Communities Index, and you can see that Dallas is largely prosperous. It has blue, there's a lot of resources, but even in near proximity, there are individuals who are living at risk places. So places that have lower consistency of business occupancy, people with lower education, lower income, higher poverty rate, adults that aren't working, and adults that don't have stability in employment. Some of the measures that go into this particular score. But just to show you, think about someone's lived experience. And even if someone has a high household income, someone could live in a more deprived area, the resources outside the home could be much lower. So with that, I'm just going to say that I think it's important that we think about the different factors that currently modify cognitive brain development. So including, like I said, memory, hippocampus, and I showed you that there is a specific role of household socioeconomic status in cross-sectional data. There's a sensitivity, and hopefully at another TACC or conference I can show you how neighborhood plays into that over time as well. And just some nuggets in case anyone else is interested. I'm not the gatekeeper. Like I said, I think it's important to think about the specificity of timing of both household and neighborhood socioeconomic status. I know some people here are doing subfield. Noah's going to collect more data It's still important to think about representative development of subfields as people work in Alzheimer's and other dementias. Think about biomarkers. Something comparable might be very important about children. Different ways we can have earlier interventions, and thinking about environmental factors that we can modify at a policy level. Should someone listen, such as something like PM 2.5, which is something that could be easily incorporated for free if you have zip code data. And this just shows you the amount of pollutants can get through the blood-brain barrier, that it can be right next to the olfactory bulbs, so it can be susceptible by just the whole brain level. Just to think about how and why individuals are different in their development as the world continues to change. So with that, I'll just thank you for your attention. Thank you, Noah and Anna, and open up for any questions.
I'd like to ask you, but I'll limit to just one. So the general observation that variance in gray matter of the hippocampus related to variance in that memory in this case,
Yeah.
generally reflected in whole brain measures, particularly in childhood. We see differences in loss of gray matter relating to relative increases in cognitive ability. Generally, in older adults, you see the opposite interaction of individuals losing more are losing more, and that's true all the way. It seems to be true all the way into early 20s if you look at aging, and yet in younger adults and adolescents, that inverse relationship of loss of gray matter relating to gaining ability seems true right up to the border of the 20s. So there seems to be some sort of there's the relationship inverts in a very short period of time. This is and this is only tangentially related to your talk, but I'm curious to get the opinions of somebody who studies individual differences in childhood as a perspective of the researchers studies individual differences in adult. On what's the mechanism? What is there a mechanistic change happening there? Is it just a Goldilocks thing? I'm curious your thoughts.
I don't think it's a Goldilocks thing. I think it partially has to do with what normal the developmental trajectories of part of the brain. So what would be normal cognitive aging versus, diverting.
Right.
So for the campus, for example, it's a non-linear change. You have volumetric increases that then plateau, and so when you're talking about that Goldilocks effects, if you're seeing that in a study of people who potentially have more volume than expected or less volume than expected range, that's where I think you sort of see these differences in the relation.
Right.
And one of the ways that people have talked about even inconsistency in findings in childhood is thinking about things like the stress acceleration hypothesis. So if earlier in life someone had a stressor that led to accelerated gain in volume, so at a certain point in childhood, they're going to look larger than someone who has a less stressful environment. What does that look like later in life? And is that it's adaptive, so we can't say it's necessarily a bad thing because it was adapting to a situation there. But then how does that play out when you're actually measuring that individual later and seeing these relations versus someone who didn't have that? Christine Waldup had 2 years ago had this really beautiful talk where she also talked about differences in intercepts, like where do people start? So is there any sort of underlying genetics that could be adaptive? So it's an unsatisfying answer to your question because I don't think it's Goldilocks. I think it's just the fact that people vary for so many different reasons. And when we have this disconnect across the lifespan, it's hard to disentangle what exactly is leading to each of those changes. Because we see these normative patterns, but we don't know why people deviate from them.
Right.
And if it was adaptive for that individual, so maybe they perform better on tasks that were important to them, or they're more aware. I don't know if that answers your question.
Well, I don't think there's a satisfying answer, but I thank you for sharing your thoughts.
Yeah.
So I'm interested in the negative correlation between SES and mentioned stress, possible source of where those differences emerge.
3 steps removed from SES, but yeah.
Pardon me?
I was saying
Yeah.
3 steps removed.
Yeah. But then you elaborated on it a little bit right now. I think stress results in further maturation patterns, slow accessing Which could lead to. And I've heard that explanation in several contexts. I'm just wondering, beyond the imaging data, what's the evidence for that?
So in animal models, you see that, you see this same pattern in mice. So mice who have greater stressors have this increased volume of glial life. So of hippocampus, yes.
Is that due, I think, is it neurons or?
Neurons and the connectivity, but it's because it's done rapidly. The idea is that it's adaptive for the time, but potentially maladaptive for the developmental cascade that would be expected.
Do you know the time scale? Is that the animal models measuring over months, or is it?
It's maybe I think 3 months, if I'm thinking, because mice are, the scale is so different, but I think it's about 3 months that you look and they get sacrificed. But it's trying to equate to around the early childhood period. So those aren't the studies that are necessarily going to what I was just hypothesizing about the 20s. But it's those early experiences that lead to variations.
And the idea is that it's an adaptive response to the stressor?
I mean, it's adapted to the environment. So if there needs to be greater awareness or memory of highly structured, emotional memories, the idea is that there's this faster maturation of the ability to remember, receive. But it's hard to ask a mouse that question, so that's just the generous interpretation back to you.
But then when you expect a relationship between the hippocampal volume and the outcomes of children?
Yeah, but I didn't test that here in that specific sample.
In the first half of the study.
Oh, for the dentate gyrus in stage 3? Yeah, you would. So that's where it gets tricky to say what are we doing. But here, unfortunately, it's just looking at one memory measure instead of the cognitive, the latent model, just because of missing data. So yeah, you would expect it to be most robust in the dentate gyrus in stage 3, but we don't see that. We do see it in speculum. I've admitted to multiple people at my poster that I've ignored speculum quite a bit because we say it's earlier maturing, it doesn't put on. But Attila Karajtash, who's in Budapest, has recently done some longitudinal work showing relations, changes in speculum and relations with memory. This is getting off of the question that you asked, but just to say, I don't think we've broadened the scope as much as we should about thinking of volumetric variations in the hippocampus and how that impacts cognition. But it isn't satisfying that where we would hypothesize it, don't necessarily see those.
Yeah, because there's these parallel sets of observations, right?
Yeah.
One about the effect of adverse exposures on earlier maturation of particular structures, the prediction that you never see that led to cognitive. In other literature, you see people living in adverse contexts to outperform those who aren't in a certain task.
Yeah.
Those two sets of things are never. I think there's a nice explanation on for each stuff.
Right.
But I've never seen that.
Yeah, and that's my hope, is to be able to more directly test that, because it's hard, right? Because we are I am not going to speak for all of you, hand waving a little bit to try to make this bridge where we don't have because I do think it's adaptive in certain situations. It's not that it's always negative, but the question that you were asking is it might be negative later in the lifespan. You don't work it.
And so just to this, and then another last question from the audience. The holy grail would be to try to identify what the mechanisms are, and those might be the same mechanisms with opposite effects. It might be different mechanisms or different sort of amount of how much you have neurogenesis and how much good neurogenesis is versus how much synaptogenesis you have, how much of deletion you have. So, and ultimately what's missing perhaps is this big interpretive framework that allows you to put it all together and then, put a sign whether adaptive.
Just one crazy idea that do studies measure spatial exploration. We know it's important for the hippocampus. We could say there are negative aspects of low SES, but there are also social aspects. I remember some neighborhoods in Montreal, low SES, kids would be outside all day and exploring and kill them after night. So the high CS would have a switch and a team. It's a caricature, obviously, but I don't measure that. there?
People measure spatial exploration and hippocampus. Jenny Pathman does that in museums at York, but I don't think it's been tightly tied to socioeconomic status together. So
So then you think that a larger volume.
of the experience, that's a positive adaptation.
Yeah.
It's an adaptation to have. No, I mean, I don't think, people have different, I try to say lived experiences because, I don't think it's that anyone who even has the same amount of resources has the same experience, right? They have better or worse social supports. And so what's interesting, it's 101, so people have to, you have these proxies, but those proxies are really just, I feel like, a good starting point, right? Of maybe looking at timing, but what's going to be more important is figuring out what specifically in a neighborhood or what specifically in a household could be increased. Because it's easier to get people to do things that they already like to do, at least, right? If you tell me to stop doing something, it's a lot harder than to keep doing something that's already working. But just figuring those things out. So, right, if kids are able to play outside more, it's an easy, but it's like, a policy decision of these neighborhoods just need more structured fields for outdoor play versus having to create their own environments. So I don't think it's too out there. I think it's important. I think it's just a part of the story of thinking of what are the differences in the experiences overall that we don't capture just looking at income and education.
Do we have any questions online?
Oh, no, we do not have any questions right now.
Thanks, Camila. In that case, Kelsey's still here for about 2 hours. So before 10, so if you have any questions, come now, and then we can still set up some time to meet. I think it really brought us together, and I can like similar projects that people have around the room. So that talk is starting those type of discussions are really great for opportunity for us in CBL to kind of share, start kind of thinking from different angles on the same type of question. So I think that's great. And with this, I'm really going to thank you for doing it for us, Kelsey.
Yeah.
And join me.